Welcome to the Cancer Research Institute webinar series, Cancer, Immunotherapy and You. Today's date is Tuesday, October 3rd, 2017, and the topic for today's webinar is Immunotherapy for Blood Cancers, an update from the field. Before we begin, I'd like to thank our generous sponsors who've made this webinar series possible: Bristol-Myers Squibb, Genentech, Regeneron, and Advaxis Immunotherapies. My name is Brian Brewer, and I'm Director of Marketing and Communications at the Cancer Research Institute, a nonprofit organization that since 1953 has focused on funding research on the immune system with the ultimate goal of developing life-saving immunotherapies for all types of cancer. This work has contributed to the development of immunotherapies, and we present this webinar series to patients and caregivers to help them understand what immunotherapy is and how it's different from other treatments, to provide information on the latest developments in research and treatment, and to connect patients to immunotherapy clinical trials. I'd like to take a moment also to mention that if you are interested in learning about cancer immunotherapy and hearing from the experts directly, as well as connecting with other cancer patients and caregivers, or exploring your clinical trial options in person with a clinical trial navigator, the Cancer Research Institute is hosting an Immunotherapy Patient Summit series in 5 markets across the United States. We've already done 3. There are 2 more upcoming Saturday morning summits in Tampa and Houston. Registration is free, and so if you're in those areas and able to attend, we strongly encourage it. Visit cancerresearch.org/summit for more information. Now, it's my pleasure to welcome today's expert speaker. Hearn Jay Cho is an associate professor of medicine and hematology and medical oncology and a treating physician in hematology oncology at the Tisch Cancer Institute of the Mount Sinai School of Medicine. Dr. Cho specializes in treating cancer patients with a form of blood cancer known as multiple myeloma and was the lead investigator on a clinical trial involving elotuzumab, a targeted antibody that is one of 2 immunotherapies currently approved for patients with multiple myeloma. His current work, through both laboratory research and clinical trials, continues to explore novel immune-based strategies for blood cancers, especially in combination with current approaches, including chemotherapy and stem cell transplants. Dr. Cho, thank you so much for joining us today.
Thank you, Brian. It's a pleasure to be here. So I really appreciate the opportunity from the Cancer Research Institute to present to you some of the latest developments in immunotherapy for hematologic malignancies. I thought it would be useful to start with an overview of normal immune functions so you understand the players on the field. And then we can discuss particular cancer immunotherapy strategies that are currently under investigation in hematologic malignancies. So the first type of cell I'd like to discuss is the cytotoxic T lymphocyte, or the killer T cell as it's known. The killer T cell is usually the cell that is thought of as the soldier or effector of the immune system. It is the one that recognizes infected cells, damaged cells, and cancer cells, and its role is to find them and eliminate them. So there's a couple of important components for the killer T cell. The first and most important probably is the T cell receptor here. Each T cell has a unique T cell receptor that is programmed to identify, for example, a virus-specific protein, and we'll show that in a few minutes. In addition, there's other receptors on the surface of the cell that helps regulate its function. So there are activating receptors such as CD28, and there's inhibitory receptors such as the immune checkpoints CTLA4 and PD1. And the decision for a T cell to kill or not kill a cell is in part mediated by the 2 signals, one from the T cell receptor and the second from a co-receptor that gives a positive or negative signal to go. And T cells contain different mechanisms to kill their target cells. One is granules. So the granules are little packages of cytotoxic material that are released upon activation. And the other is through surface molecules that send a death signal to the target cell. The next type of cell I wanted to talk about is the natural killer cell, or NK cell. NK cells are similar to T cells, but they have slightly different functions. So natural killer cells do not have a T cell receptor, obviously, but they do have something called an Fc receptor. Fc receptors recognize antibodies, particularly ones that are stuck to infected or damaged cells. And similar to T cells, NK cells have granules which they release to kill their target cells. Next component I wanted to discuss is monoclonal antibodies, or antibodies in general. There's a number of immune therapies that consist of engineered monoclonal antibodies or humanized monoclonal antibodies. You'll hear that terminology. But a monoclonal antibody is simply an antibody, a single type of antibody that is in its pure state and used typically as a treatment option for cancer. Antibodies have two parts. There are two antigen binding fragments, or FABs. This is the part that recognizes the target on the target cells. So these can recognize virus proteins, bacteria, and a variety of other targets. And then there's a third part called the constant region, or FC. And this is the part that talks to the rest of the immune system. So as I mentioned earlier, NK cells express FC receptors, and they interact with the FC of the antibodies. So how do all these components work? Well, under normal circumstances, you have a normal cell, which is perfectly healthy and happy and has nothing wrong with it. Normal cells are constantly recirculating bits of their own proteins on the surface on a molecule called the major histocompatibility complex, or MHC. So you can see here that these red, yellow, and blue dots represent self-peptides, or normal proteins, that are being recirculated on the surface of the cell to be examined by passing T cells. So as I mentioned earlier, the T cell receptor on a T cell here scans through proteins that are being displayed on the major histocompatibility complex here. And it reads it like a barcode. So each T cell looks at the peptide in the MHC molecule and sees whether it's a self protein, which it does not recognize as foreign, or if it's a virus protein or some other type of abnormal protein. So in this case, it recognizes a self protein, and that's a red light. In addition, healthy normal cells express inhibitory receptor ligands. For example, PD-L1. This is a so-called immune checkpoint. And this interacts with PD-1 on the T cell, and that's an inhibitory receptor, so that also gives a red light and prevents the T cell from killing this normal cell. So what happens in the opposite situation? So here's a normal cell, but it's going to be infected by a virus. And that virus makes virus-specific proteins. So these are not normal proteins. And these proteins get expressed on the surface of the cell in the context of MHC. And again, the T cell will scan through. In this case, it sees this virus protein, and this T cell happens to be specific for this particular virus protein. And it, in addition, infected cells downregulate surface molecules such as PD-L1 that would normally inhibit T cell activation. So in this setting, the T cell scans through the peptide, and it reads it, and it reads it as foreign, so it gets a green light. It wants to attack this cell. And there's no negative signal here from the PD-1, PD-L1, and as a result, the T cell will kill this virus-infected cell. Similarly, if a normal cell is infected with the virus, it may express virus proteins directly on the surface. This is independent of MHC. And there are antibodies that recognize those proteins, and then they'll stick to them. So these are normal antibodies produced by your immune system when you were vaccinated or infected with a virus. And those antibodies stick to these foreign virus proteins on the surface of the cell. And then an NK cell moves in, and through the FC receptor, it interacts with the antibody and gets the go signal to kill that virus-infected cell. So, to summarize, cytotoxic T lymphocytes survey the cell for non-self antigens or proteins and kills infected cells. It also can kill abnormal cells, such as tumor cells, under specific circumstances. And then antibodies stick to cells expressing so-called non-self antigens and mark them for immune destruction. And I'll show you in a minute now how we can use these mechanisms to develop cancer therapeutics. So there's four major classes of cancer therapy strategies that I would like to discuss today. The first is targeted monoclonal antibodies. So these are engineered monoclonal antibodies that mark tumor cells for immune destruction. There's tumor vaccines, which train T cells to recognize and kill tumor cells in the same way that, for example, an infectious disease vaccine trains your immune system to resist infections. Engineered T cells, particularly the chimeric antigen receptors, which add to the patient's T cells. We add chimeric antigen receptors to patient T cells to target tumor cells. And then finally, immune checkpoint inhibitors, which are monoclonal antibodies designed not against a tumor target, but to block negative immune signals, as we discussed earlier. So how do these work? So this is an engineered monoclonal antibody right here. There's a couple of examples that are already approved by the FDA, and they recognize tumor markers. So for example, CD20 on the surface of lymphoma cells or CD38 on the surface of myeloma cells can be recognized by a monoclonal antibody. For example, rituximab is an engineered antibody that recognizes CD20. Daratumumab recognizes CD38. These are approved by the FDA for their respective diseases. And these antibodies will stick to the surface of the tumor cells and attract NK cells. And the NK cells will then kill the tumor cells. Tumor vaccines are similar in strategy to infectious disease vaccines. So you have to identify a unique or relatively unique cancer antigen or cancer-associated antigen. This is a protein that is relatively unique to the cancer cells. And these proteins can be purified and produced in a vaccine form. And in this strategy, patients will receive repeated vaccinations to stimulate both T and B cell immune responses. And in this example, we're using a protein called MAGE-A3, which is a well-studied tumor-associated antigen that happens to be expressed in multiple myeloma. So the vaccination strategy will stimulate MAGE-A3 specific T cells, which can recognize MAGE-A3 that is expressed by tumor cells and kill them. In addition, MAGE-A3 specific antibodies may be generated that help build immune responses by recognizing the protein when it's released by tumor cells and helping to train the immune system to recognize it further. Engineered T cell therapies, particularly chimeric antigen receptor cells, are getting a lot of publicity now, and for good reason. In this strategy, we remove patient T cells in bulk, and then we introduce a gene into those cells. So this is an actual gene engineering therapy, which inserts a gene for a chimeric antigen receptor into those cells. So now all these T cells express the same receptor, and that receptor can recognize a tumor target on the surface of tumor cells. So in this case, for example, the chimeric antigen receptor, or CAR, for CD19, which was recently approved by the FDA. So this tumor-specific CAR recognizes CD19, and once the gene is inserted into these T cells are expanded outside the body in the laboratory, and then they're reinfused back into the patient. And those T cells now are hardwired to recognize the target, in this case CD19, on the surface of the cancer cells. And those T cells can find and kill cells that express the target. And then finally, immune checkpoint inhibitors function by releasing the brakes on the immune system. So as we saw earlier, T cells recognize targets on the surface of tumor cells. In this case, it's a tumor-associated target. And it reads it, and it wants to, it recognizes that antigen is being foreign or tumor-associated. But tumor cells express inhibitory receptors such as PD-L1. So that can give a red light and block the activation of that T cell and prevents it from killing the tumor cell. Now, immune checkpoint inhibitors, which block those negative signals here release the brakes. So it removes that negative signal that turns off the T cell immune response and allows that T cell to kill its target cell. And as many people here know, the immune checkpoint inhibitors have been successful in solid tumors and have been approved by the FDA for a number of different tumor indications. So what's the progress in hematologic malignancies? There's been a lot, actually. Both nivolumab and pembrolizumab, which are immune checkpoint inhibitors that target PD-1 on T cells, have been approved for relapsed classical Hodgkin's lymphoma based on clinical trials in this disease. There are two targeting antibodies, elotuzumab, anti-SLAM-F7, which was approved in combination with a drug called lenalidomide for relapsed multiple myeloma, and then daratumumab, which is an anti-CD38 monoclonal antibody, which was approved alone and in combinations for relapsed multiple myeloma. And they join rituximab and other CD20 targeting monoclonal antibodies as FDA-approved agents in hematologic malignancies. And finally, as recently publicized, the anti-CD19 CAR T cells were approved for ALL, or acute lymphoblastic leukemia. So there has been a lot of progress in this area, and there's a lot of optimism for immune therapies in hematologic malignancies. However, there are challenges. So for targeting monoclonal antibodies, the challenge is finding the best tumor-specific targets in each individual blood cancer. For tumor vaccines, similarly, we need to find the best tumor antigens and vaccine formulations to stimulate a strong anti-tumor immune response. For engineered T cells, such as CARs, we need to find the best targets for the CARs and also where to use them. Is this a strategy that we can use upfront for newly diagnosed disease? Or because of the potential risks and benefits involved, should we reserve this for patients who've relapsed after conventional chemotherapy? Immune checkpoint inhibitors, what are the best inhibitors? And when should we give them? Again, the question of whether this is something that can apply as an upfront therapy for relapse disease or for refractory disease. And in my opinion, what the biggest question is, what are the best combinations of immune therapies? There's a lot of success for individual therapies in each disease, but the response rates in some cases are relatively low. And it suggests that by combining different immune strategies, we may be able to increase the efficiency of mobilizing the immune system so that they can kill cancer cells. So these are all important questions that researchers in the field are trying to address both in the laboratory and in the clinic. So what we're looking for, what we're hoping to see is a future where we can combine immune therapies with other types of chemotherapies, including chemotherapies, so-called targeted therapies, in some cases maybe radiation or surgery, to induce remission of blood cancers, and then using strategies such as vaccines, CAR T cells, and immune checkpoint inhibitors to stimulate strong and durable anti-tumor immunity with the ultimate goal of broadly applicable curative therapies for some of these blood cancers. So that's our mission, and that's the goal of our research. And with that, I thank you for your attention.
All right, are you able to hear me now?
Yes,
Great.
I can hear you, Brian.
Well, thanks so much for walking us through some of the exciting progress that's being made in the treatment of blood cancers, and especially multiple myeloma. At the very beginning, we mentioned that a major focus for your research is on combining immunotherapies with other forms of treatment like chemotherapy, and you closed with that too. So I was wondering if you could just say a little bit more about how those two things would work together. First, how does a chemotherapy work, and how is that different from the different mechanisms of action you showed us with the immunotherapies? And then how do those two things synergize?
So that's a good question. So most chemotherapies at a very broad level are toxins, they're poisons. They poison various processes that occur in cells, and the intent is that tumor cells are more dependent upon certain processes, such as replicating their DNA or dividing quickly. And by attacking those processes, you can kill tumor cells efficiently. Unfortunately, most of those functions also occur in normal cells, maybe at a lower rate or less frequently, but that results in side effects. So there are really very few chemotherapy agents in the conventional sense that are truly selective for just the tumor cells. On the other hand, some of these chemotherapies are very good at killing tumor cells. So, for example, in multiple myeloma, we have combinations of chemotherapies that are generally fairly well tolerated and in most cases can induce remission in multiple myeloma. And so we have established therapies that can reduce the disease burden to a significant degree. And it may, if you think about, for example, bulky tumors or widespread disease, it may be difficult to stimulate a strong enough immune response to eliminate large numbers of tumor cells, whereas we have chemotherapy strategies that work pretty well and in many cases are pretty well tolerated. So one could imagine using conventional chemotherapy treatment to reduce the amount of disease, to render a minimal disease state. But then going in with an immune therapy such as a vaccine or a CAR T cell or an immune checkpoint inhibitor or some combination of those things in order to stimulate a strong immune response that can go and find those residual tumor cells and kill them. So that's one scenario. We are investigating that in myeloma in the context of what's called autotransplant, which is a standard therapy using conventional chemotherapy. But that does render a minimal disease state in most patients. So making that type of strategy better by adding immune therapy to it is one approach.
And so the chemo goes in and does kind of a blanket, kills the cancer cells very rapidly, and then the immune system comes in to kind of sweep up any remainders that might be crawling around in the blood, as well as to patrol for any future developments. Is that a good analogy?
Right. So that's one general approach that we're interested in, and that's basically the hypothesis that we're investigating.
You mentioned vaccines, and in the specific example, you showed the MAGE-A3 vaccine, which is a specific type of antigen that's part of a family that has been the focus of vaccines that are kind of off the shelf. They're found in multiple people. But a lot of buzz lately is the word personalized vaccines and neoantigens. And I was wondering if there's any work being done in multiple myeloma or other blood cancers around that personalized, and what does that mean for a patient if a patient hears this in the news?
Right. So that's a good question. There's two general types of tumor antigens or tumor targets for immune therapy. One is like MAGE-A3, the so-called off-the-shelf antigens. These are proteins that are produced in tumor cells, but they're abnormal expression of normal proteins. So these are proteins that are not normally expressed in, for example, plasma cells in myeloma, but in the malignant plasma cells of multiple myeloma, they're expressed. These are proteins that are turned on abnormally in tumor cells. And the advantage is, is that they're the same in most people, right? So if you had, for example, a vaccine that targeted something like MAGE-A3, you could potentially use it in any person whose cancer expresses MAGE-A3, right? And so there's certain advantages and disadvantages to that. On the flip side, there are certain types of truly unique individual patient-specific targets. So for example, most tumors have mutations in them. So mutations are damages to the DNA that changes the code. And in most cancers, those types of mutations or damage to the DNA causes expression of abnormal proteins. In other words, proteins that are not the same as the normal protein because the code has been changed in the DNA. And in some cases, the code changes enough so that the protein that is produced by that gene is significantly different from the normal protein. And like a virus protein, the immune system can recognize it as foreign. And there's evidence now from a lot of clinical trials, for example, with immune checkpoint inhibitors, that at least one of the major mechanisms by which immune checkpoint inhibitors work is it unleashes T cells that recognize those mutated proteins. So we call these mutated neoantigens, so new antigens that are created by mutations in the DNA code of the tumor cell. So the tumor cell's DNA is different from normal DNA, and as a result, there's the production of abnormal mutated proteins that can be recognized by the immune system. So since we know that the immune system can recognize these abnormal proteins, we're taking it one step further. Can we increase the number of T cells that recognize those types of mutations by vaccinating people against the mutated protein? In other words, can we mobilize an immune response against the abnormal protein by stimulating the immune system over and over again with a vaccine? So there's a number of clinical trials taking that exact strategy. There's a couple underway in myeloma. So the way that works is when a patient agrees to participate in the trial, after they give their consent, we take a specimen of their tumor. So in myeloma, it grows in the bone marrow. So we can take a sample of the bone marrow, filter out the myeloma cells, and just sequence the DNA from that tumor cell. And then we get all this information, and we can identify the mutations that are in the DNA of the tumor cell from an individual patient. And then we can analyze that using a variety of computer algorithms to identify which mutations are most likely to be recognized by the immune system. And then we can make synthetic peptides, so proteins can be made in the laboratory and used to vaccinate patients to stimulate their immune system to recognize those abnormal proteins. So the plus side is these are proteins that are only expressed in the tumor. So it's a truly individualized tumor-specific approach. So if you do mobilize a strong immune response against the mutated protein, it'll only kill cancer cells. It will not kill normal cells. The downside is it's a very difficult and labor-intensive process for each individual. We have to do this for each individual who undergoes this type of treatment. So the peptides that work for one person are not going to work for another person because they'll have different mutations. So there's advantages and disadvantages. The clinical trials are underway, and we're going to understand how well this works and how we can apply this in different types of blood cancers.
That's wonderful, and it's something that I know that we at the Cancer Research Institute are very interested in, and there's a lot of work going on in that area. So hopefully more good news coming from that sector in the near future. So, you know, like all cancers, blood cancers they're diagnosed at different stages in terms of how far along the disease has progressed in a patient. At what point is there a certain point when a patient should ask about immunotherapy, or is that something that should be part of the discussion at the very beginning? Where in the treatment plan does a patient begin to encounter immunotherapy, or how do they do that?
That's a good question. Because of the process of approvals for different types of drugs and agents in cancer, typically a new immunotherapy agent will be approved for relapse or refractory disease first. And then the sponsors or companies or not-for-profit organizations or consortia will conduct clinical trials moving that therapy earlier and earlier, for example, into upfront initial treatment to determine whether that works or not. So the best example that we know is rituximab, which was originally approved for relapsed lymphomas, but as that process was underway, clinical trials were being conducted moving that adding Rituxan to standard upfront chemotherapy for lymphoma, and it was shown to be superior to older treatments. And as a result now, Rituxan is part of the standard upfront treatment for a lot of different types of lymphoma. So if you are diagnosed with a blood cancer and there's an immune agent that is approved for relapse and refractory disease, but there are clinical trials underway moving that earlier in therapy, it is certainly worth your while to investigate whether a clinical trial option is available, because there's at least the possibility that moving that immune therapy earlier in treatment may result in better outcomes for patients who get it earlier. Alternatively, if you've already been treated for a particular blood cancer and you're experiencing a relapse, you should speak with your physician about clinical trials for relapse disease using immune strategies. For example, CAR T cells are very hot right now for different types of hematologic malignancies. There's a number of different immune checkpoint inhibitor trials underway and a number of different vaccine trials underway. So there's all these options are either under investigation, or in some cases there are FDA approved immune therapy options for relapse disease.
So we couldn't do this webinar today without me asking a question about something that's been in the news recently around the FDA putting a clinical hold on trials testing checkpoint inhibitors in multiple myeloma patients because of some deaths that were reported in those trials. Can you say anything about what happened, or what do we know may have happened, and why are multiple myeloma patients seemingly more susceptible to this risk than patients with other types of blood cancer?
That's an excellent question, and I think what it shows first and foremost is that the system works. The FDA recognized that unexpectedly in 2 clinical trials for immune checkpoint inhibitors in multiple myeloma, the patients who received the standard treatment plus checkpoint inhibitors had higher rates of death compared to patients who received standard therapy alone. And clearly this was a cause for concern, and the FDA took an appropriate approach to first put a hold on those 2 clinical trials to review the safety data, and as a higher level of precaution, given the relative newness of these types of treatments in myeloma, to hold all clinical trials involving combinations of immune checkpoint inhibitors with certain types of myeloma drugs in order to ensure safety for the patients. So at the base level, it shows that the regulatory process is vigilant, and we are putting patient safety first, as we should. The second question is more difficult to answer because we don't have a lot of information. What we do know is that there were increased numbers of deaths in the immune therapy arms, and that some, but not all of those deaths could potentially be attributed to the immune therapy. So we didn't discuss a lot of the details, but immune therapies have as a major side effect, they can cause autoimmune responses, which are adverse. So inflammation in the gut and the skin, fevers, and other side effects that are caused by abnormal recognition of normal tissues by the immune system. But not all of the deaths in the immune treatment arms could be attributed to those types of complications. So it's really not clear why in those 2 particular trials there was a higher rate of death. In addition, there's probably at least a dozen other clinical trials of different combinations of immune checkpoint inhibitors in multiple myeloma, and the safety reviews for those trials have not revealed higher rates of death yet. And as I said, there's a broad review of all the safety data that's underway, but most of these clinical trials have been open for a year or 2, and there are built-in safety review points for those clinical trials, and none of those other trials were purporting unexpectedly high death rates in the treatment arms. So it was appropriate to take the safety precaution in this setting, and I think that was very important. And the appropriate review of the available information is underway. And I fully expect that in the next weeks or months, hopefully sooner, that the FDA will come to a determination as to how to best approach this. Do we add additional safety safety measures into the trials that are already underway? Do they need to be modified in some way to ensure that patients are safe? And I hope so, because I think there's a lot of reasons to expect that immune checkpoint inhibitors will play an important role in immune therapy for myeloma. And we would like to continue our clinical investigation into this area, but we want to do it safely and to ensure that our patients are not exposed to excessive risk in this setting.
Well, I'm glad.
So we're hopefully going to get more information and get those trials back underway.
Good. Well, I appreciate that. That clarified things. Actually, I was going to ask you, were the deaths immune-related adverse events from the therapy, or was it just disease progression? So it sounds like it's not entirely across the board, and that most importantly, as you said, the system works in terms of how regulators govern safety and protect patients from risk. And just on that note, one of the things that we do a lot at the Cancer Research Institute is inform patients about their clinical trial options and connect them to clinical trials of immunotherapy. And as part of that process, that can be very, it requires demystification, right? There's a lot that many patients don't know. Many of them have never even heard the term clinical trial before. And so explaining to them that what a trial is and how it fits within the research spectrum and the role that they play is super important. So my question is, I don't want patients to feel that they need to be afraid of entering a clinical trial. So in your own practice, when you bring patients onto trials, how much do you walk them through all the risks, potential risks and potential benefits?
So it's important to recognize that any treatment, no matter what it is, an aspirin, a Tylenol, an antibiotic, a chemotherapy drug, an immune therapy drug, there's no such thing as a no side effect drug. Every potential therapy has some side effects, and some people experience no side effects, some people experience all the side effects, and everything in between. But it is important to understand those risks. So for example, if a patient comes to my office and they have multiple myeloma and we are discussing their treatment options, we need to recognize that for any given treatment for this patient, there are risks. So risks of side effects of drugs, risks of delaying therapy, or risks of doing therapy that's not effective. And then there's potential for benefit, right? And you want the risks to be relatively low and you want the benefits to be high. So we discuss each option sort of at a very broad level. So standard chemotherapies, we have very well defined risks and we have a good idea of what the benefits are because there's statistical data. And then for clinical trial options, there's really two kinds of clinical trials. There's clinical trials that include new combinations or new applications of drugs that are already approved by the FDA, right? So new combinations, new applications, we have a pretty good idea of what the risks are because this is a drug that's already been through the regulatory process. We know what the side effects are. And the benefits, even though in that particular context we may not know exactly what the benefit is, we do know that that agent has a certain amount of benefit in a given disease. And then the second type of trial are trials for new drugs, where we may or may not have a lot of information about risks, and we may or may not have a lot of information about benefit. And it's important to have clarity when we're discussing these risks and benefits with our patients. It's not helpful, I find, to go through the list of every single potential risk that has been reported by anyone who's ever been exposed to this drug, because it's too much detail and it's not really helpful. It's information, but it's not knowledge. So what I like to convey, what I try to convey to my patients is what are the expected risks, in other words, what are the most common side effects, and what are the most serious side effects. It may be very rare, but it may be life-threatening. And so that we understand what the risks are going into either a clinical trial or standard therapy. And then the second part, which is important, is what are the potential benefits, right? If we have a high expectation of benefit, that may justify a higher level of risk, for example. And then a lot of this also is related to where the patient is in their disease and what the patient's attitude is towards treatment for the disease. So ultimately, it's the patient's decision, and we do our best to provide the necessary information so the patient can make a decision with confidence.
Thank you. And I know we're almost out of time, but we did receive a few questions about chronic myeloid leukemia and what immunotherapies or trials are available for that. Do you have anything that you can add to that?
So CML is actually a good example of a disease that's benefited both from historically from immune therapy and also from the development of new drugs. So CML was treated in the past with allogeneic stem cell transplant. So this is a stem cell transplant where a donor provides stem cells to a recipient to engraft a new immune system. So allogeneic transplant is actually the first immune therapy for cancer. And so the graft develops a new immune system that can recognize the cancer cells as foreign, and there's what's known as the graft versus tumor effect, where that new immune system finds and eliminates tumor cells. So that has become less common in recent years because of the development of new drugs, particularly what are called targeted kinase inhibitors, which have very high response rates in CML. And even if it doesn't completely eliminate the disease, it can keep it under control for years or decades. So there's not as many transplants being done for CML, but it does relapse even with the contemporary chemotherapy agents. So since CML can be cured by immune therapy in the form of allogeneic transplant, there's a very good, sound rationale to explore some of the modern immune therapy strategies in CML. So there are a number of different clinical trials underway for relapsed CML, investigating these types of immune strategies. And I would certainly encourage anybody who has CML that has relapsed after kinase inhibitor therapy to discuss with their doctor what their options are for clinical trials for immune therapies.
All right. Well, thank you so much for joining us today. I know you, like many doctors, are very, very busy, so it's very precious for our audience that we get to have this time with you today to talk about immunotherapy breakthroughs in blood cancer. And so with that, I'd like to close by once again thanking our generous sponsors who've made this webinar series possible. Also like to remind you again of our Immunotherapy Patient Summit. We have summits in Tampa and Houston coming up. We also very shortly will have recordings of the New York City Summit that took place earlier in September available. So if you're unable to attend the summit in person, you can watch online. So again, that's cancerresearch.org/summit. Also, the Cancer Research Institute website provides information about immunotherapy for a variety of cancers up to, I think we're up to about 20 or 21 different categories of cancer now. And this number is growing rapidly as immunotherapies enter more and more different types of tumors. So if you are interested in learning about immunotherapy either for one of the blood cancers we discussed today or any other type of cancer, go to cancerresearch.org/patients to browse our patient information resources there. And with that, I'd like to also say our entire webinar series is available on the Cancer Research Institute website and on our YouTube channel. The quickest way to find it is to go to cancerresearch.org/webinars. Dr. Cho, once again, thank you very much, and thank you all for watching today. Goodbye.